presentation would be overview of the ongoing collaboration within CompBioMed and the DICE project on the data transfer workflow developed there. Be presented by Narges Zarrabi and Alistair, who also is connected from UCL, sending updates.
Yes, thank you, Marco. Good morning. I'm Narges Zarrabi from SERP, and I'm giving this presentation, presenting the collaboration with DICE, CompBioMed, together with Alistair Smith from UCL. Alistair, are you there? Can you?
Hello, yes, I'm here.
Great, okay. So basically we have been setting up this collaboration with DICE somewhere beginning of the DICE project. So we submitted a proposal together with them, and we got some grant to help CompBioMed to make use of DICE services and EU.dot services. And today I'm going to give an update on where we are. Before talking about that, I would like to say a few words about the data management hackathon we had, combined with the CompBioMed All Hands meeting. So we were here in this small room the day before of the CompBioMed All Hands meeting, and had a very nice session about data management, nice discussion about metadata. And I hope this is going to be useful for the upcoming task on data curation and deliver the upcoming deliverable we have on that. So luckily some people from CompBioMed were there, and I hope this has been useful. We had experts from data management telling us about metadata and metadata schema. So I think it was a great good session. The reason we set up this collaboration is, of course, the common data management challenges that research communities, including CompBioMed, face. And well, it's all about efficient data access, data sharing. We also had it yesterday about importance of being easily share data, collaborate on your data. Intensive data sharing, especially for communities like this, which the research is really compute intensive and also data intensive. Restricted data sharing and data transfer. In this whole data management, we want to preserve data, so even in 10, 20 years, the data can still be used, find the data provenance, where has the data come from, what has happened to the data that is in this final shape. And also making research data accessible, making sure there are persistent identifiers or DOIs attached, and they are findable by other research communities. So one of the workflows or use cases that we started with within CompBioMed was a workflow using the ALI application for computational mechanics. They created, it was used by, Jasmine and her team in the Barcelona Supercomputing Center. So they came to us with this problem that we generate a lot of data in a lab, and they needed to transfer the data to the Barcelona Supercomputing Center. And they did a lot of pre-processing on this data, image stitching, segmentation, to make the data ready for simulations. And the idea was to make replicas of this data in the other HPC centers, including SURF or EPCC or LRZ, because this ALI application is also installed on those supercomputing or HPC centers. And the data can be used by other researchers, for example, a team at UVA or in other research teams to also run simulations using this application on the data. So they needed to transfer a large amount of data. And that's what we call data replication. And this is also important to have replicas of a data that you have in other sites. If something happens to the data at BSC, there is still one copy of that data at SURF, for example, and that can be replicated back. And once the data is replicated, of course, researchers can run simulations and do analysis and come up with interesting output. So this was a typical use case that we started with to set up this platform that I will show you in a couple of slides. We focused on implementing two workflows: a data replication workflow, which means replicating a big data set between different HPC centers. And it doesn't mean that it only makes replicas, but it also means that once you make a replica, for example, the B2 safe at instance at Surf is connected to the data archive in the backend as storage. And it means that the data will land on tape medium, and it's meant for preserving data in longer term. So the data will be preserved, but also made available to other centers and other researchers. And the other workflow was data publication workflow to make sure that a data set will be ended up in a data repository published with metadata attached. So in this collaboration, three main partners were involved: UCL, BSc, and SURF, but we also had LRZ and EPCC involved as, let's say, supporting. And we used three services of EUDAT, the B2 share, which is the data repository, the B2 handle, which is for making persistent identifiers, and the B2 safe, which is the middleware based on iRods for actually replicating the data. So again, the EUDAT services that was used and some resources that was planned to be allocated. So the B2 share, the idea is that UCL, which Alistair will show later, is going to deploy an instance of B2 share data repository connected to storage at the UCL up to 50 terabytes, so that if you want to publish a large data set, you can always publish it there. There is also a B2 share community instance, but that is not meant for publishing large data. Then the B2 handle, so Surf already has this. UCL also got a B2 handle through the DICE virtual access mechanism, and BSc also, for making PIDs and assigning it to the data. So once a data set is replicated from BSc to Surf, with this B2 safe service, there will also be PIDs assigned. So making sure that the data will later be can be made findable. And the B2 safe service, of course, which is a heavy service to deploy on an HPC center. It's not meant for a research team to deploy it, but at the HPC center. But then that center can help out researchers who want to collaborate with other sites and transfer large amount of data. This is the setup or what we are trying to set up with this collaboration. As you see, the main centers or partners are listed: the UCL, SURF, and BSc. BSc is a compute site and a B2 safe endpoint. They have deployed the B2 safe. So if you are doing any research at BSc and you want to transfer data to Surf, or you can contact the technical admins of the data management at BSc and, well, have it done, because Surf is also a B2 safe endpoint, and Surf and BSc are federated and tested. So you can shift large amount of data as we speak. As I said, Surf is also an archiving site, so any data that is replicated to Surf will be end up on tape, meaning that it is preserved for longer term. And it's also possible to publish data at Surf data repository. So this is a similar B2 share service that we have at Surf, meant for publishing large data. And you can publish your data again through this DICE virtual access mechanism, so via the EOS portal, as I showed yesterday. And UCL is the data publication site, so they are going to deploy this B2 share data repository connected to the storage at their side but also decided to deploy a B2 safe endpoint, meaning that data can be replicated between UCL to the other side. And this is, I think, a very good thing, because I know that a lot of research is being done at UCL and the research team there. So I think this is a great opportunity that the team of James and Alistair are a great effort that they are putting into this to set this up, and it will later help researchers to shift large amount of data between centers. So we started with a work plan with some timeline and a bit of, well, started with deploying things. It took some time to do preparations, but status of the task so deployment of B2 Safe at BSc is done. Also B2 Handle, they have a prefix. They are now able to provide make PIDs, because this is basically required for the B2 Safe. Federation with Surf, well, I've put still on progress. It's there, it's tested, but we are still going to do some performance tests. Unfortunately, I couldn't get those numbers from the technical teams, but we are now busy doing performance tests to get an idea. Okay, I should speed up. I think I'll, well, this is a future concept, so we would like other HPC centers at CompBioMed to also be connected to this network, such as LRZ, which is already federated to SURF and EPCC, and hopefully make a network of HPC centers that can collaborate and transfer data easily. Well, I will hand over to Alistair to talk about his part. Alistair, are you there?
I am, yeah. I've realized that possibly this slide is now a little bit redundant in the context of the ones you have shown, but as said,
okay.
we're implementing B2Share and B2Safe, and B2Share is for publishing data on a web portal, and B2Safe is about federating and distributing data. For the next slide, yes, okay, I have my own set as well. Okay, so as far as UCL interest in these technologies, so we're doing it to support the CompBioMed project, which is ongoing, but also UCL has its own interest in publishing and preserving data. And so part of this is about exploring the technologies, seeing how they work. In particular, like compared to, so we're using Figshare for institutions for publishing data right now. But we don't have so much control over the schema, and it's interesting to see the more flexible approach that's in B2Share. The next slide. This is actually mostly created by our networks team. So UCL is quite big, and we have different departments, and it's to do with the connectivity of what we're doing to the outside world and how to protect and safeguard it. So most of this, these gubbins are really there for B2Share. As a web application, it's a bit that could be connected to by anybody, basically. It's somewhat vulnerable. So the place where people connect is in the top left. So for B2Share, you've got remote users from the internet, and they'll have to go through a firewall, which a web application firewall, which is expected to be Cloudflare. And the B2 Safe traffic, although it looks like it's going to go through that, actually the diagram slightly misrepresents things. That's going to go straight through. So we've got a couple of firewalls within the university that it needs to go through. After that, B2 Safe will more or less just be allowed through based on IP ranges. And then we have a little green icon, which is a piece of technology that might well wind up being another piece of technology. But this is basically a load balancer, traffic manager, and most network routes from the outside world go through this at UCL. And at the bottom, we've got the thing that's really the bit that I'm working on. So there's a DICE virtual machine, which is going to be running both B2 Safe and B2 Share, unless it turns out that we have to spread this out. And that's connected to some storage that we operate. This is production storage. We've annexed off a part of it for this purpose. So that's NFS mounted. And for B2 Share running on this DICE virtual machine, the authentication is going to be through B2 Access. And next, oh, hang on, do I have anything else to say there? No, that's good. Yeah, so on this slide, so this is a combination of the our implementation and the required software stack. This is for B2 Safe, and most of my work so far has been on B2 Safe, so I won't have much more to say on B2 Share. So starting at the top, we're providing 50 terabytes of storage, which is using a file system called Spectrum Scale, formerly GPFS, and that's backed up every day. Currently, it's backed up onto Spectrum Protect, which was formerly called TSM, and in the future, it will probably be something called Cohesity. So we have an NFS mount going from that to the virtual machine that I'm running this all on. That's running Red Hat Enterprise Linux 7, and it's a VMware install. There's a separate partition for the database, which you will see is needed for this, for running iRods. And those partitions are backed up onto Cohesity also. So on this virtual machine, we've got PostgreSQL 14, a database, not necessarily that version, but a database is required for iRod server. And that's a requirement for B2 Safe. And we've got those all installed. Let's see. Okay, so there's another bit. Part of the way that B2 Safe federates its information, performs its federation to know how where the original record is from a copy that might have been distributed between a few sites. It's information stored in PIDs. And we could have run our own PID server for this, but we decided to use the services of Surf, who already have this functionality. So there's we needed the B2 Handle client, but that's communicating with Surf to provide that functionality. So this is basically where we are now. I've run a set of tests and done a bit of troubleshooting. And B2 Safe seems to be working as a single instance properly now. And so the next stages are to federate with other institutions, which are expected to be Surf and BSC. So final slide. Oh, sorry, final for me. It's not quite the final one. So just to talk about some of the challenges we've had. So for B2, so this is really all for B2 Safe. The installation information is spread over a few different sites. So there's some on the EUDAT site, there's some on GitHub, some on GitLab. The documentation is okay, but there's bits missing. I suppose this is temporary. Right now, one of the required repositories just doesn't work, and there seems to be a broken dependency, or there was a broken dependency when it was still working. The support is limited. You know, people have other priorities as well. Oh, so there's a sort of interesting thing to do with the way that UCL works, but basically at UCL, if you want a virtual machine and you want it to be centrally supported, then you've got a choice of Windows or Linux, and if you want Linux, then it's Red Hat 8, and that doesn't work with the current software stack. I put a lot of effort into making Red Hat 8 work, and that ultimately didn't work. So I was able to get a Red Hat 7 install, and this works. This is compatible with iRods, but that was a fair challenge earlier on, and that's. Oh, it's a small thing, but at least last time when I was installing this, B2 Safe isn't compatible with the latest version of iRods, and just from a perspective of running automated updates, then you need to be a bit careful to make sure that you've got the correct version set up. So that's just, yeah, those are the main issues.
Thank you very much.
That's me done.
Oh, thank you, Alistair. I think these issues that you have listed here is also a very good maybe feedback to the DICE and to the B2 Safe core team, because I know that they are eager to know about this and do improvement and work with communities. So I think this has been great work. Thank you for presenting. And this brings me to the last slide. So if there are any questions, I'd be happy to take.
So Alistair over there is essentially working, I think, with James Wilson, and James has been in information services. What's interesting here, I've repeated several times in the past, that you hinted at it, this has become of interest to UCL as a service for everyone. It's a huge place, and if this will be made to work, it has to run in a professional way and persistently, rather than here today, gone tomorrow, because it's just a project that has a finite duration. So getting this to that level is an important part of it. It's stress test stuff in a different way. Do you agree? Rather than if it's just a CompBioMed 2 thing, well, CompBioMed 2's only got 12, 15 months to go. And if it doesn't match services there, maybe it drifts to something somewhere else. You see what I'm getting?
Yeah.
It's an academic effort that is best efforts, but is it professionally robust?
Yeah, I think
it's good that ISD information services people like this and want to make it.
Yeah, I think this is a very important point, because it's not bounded to the end of the project, because the ISD department, which is the IT, I think, department of the whole UCL is involved. And the idea is to have this running as a service, independent of different projects that can serve different projects and different research.
But just to say really quickly, I've had a lot of engagement with ISD, and it basically is the commoditized end of our IT. And it's a giant leviathan that does things on an institutional level. So it has to have a standard of ease of installation and use and ease of support that would allow it to be brought in at enterprise level. We always moan about, you can't do your own little thing, but getting an enterprise to adopt something as a commoditized platform is quite exceptional. And it really does need that kind of ongoing assurance that things would be in place.
But let me just add to this, because you know what I mentioned to you yesterday as well. UCL has very recently created a new entity called ARC, which stands for Advanced Research computing center, and it's come out of ISD, but to support research, which is very different from the notion she was using at Andrea Enterprise for everyone, students and whatnot. So this is really trying to do something in an academic context that's informed by using this expertise from ISD, but focus on supporting researchers, which is quite good because it then brings the things closer together, right? So it's important for us that this is sustained.
Yeah, no, this, and I saw the arc in the beginning of Alistair's slide. I was not aware of this.
Yes, it's really created in the last few months. I've become an associate director of it.
Yeah.
And it at the moment has quite a lot of potential to do interesting things.
It's also a good way to get underrepresented demographics on, so the social and historical sciences, the arts and humanities, because when you put it across an institutional level, people start talking about it, and then you suddenly realize someone has access to something that you not known about, and it's a really good level of equalizing.
Yeah, no, this is very good point, and there are other institutes or centers which are have set up like SURF has set up similar services. So this exchange of knowledge.
In fact, I'm coming to see the people at SURF middle of July as representing ARC, where its interest and all, and this is one of the topics.
Okay, no, very interesting. Okay, thank you.
Thank you. So, yeah, we can move to the last presentation before the coffee break. Alex Wade from UCL will present about the portability and scalability of MD code. So, Alex, 15 minutes.
Thanks. Morning, everyone. I'm Alex, and I'm going to talk about some of the work we've been doing in Peter's group on binding affinity calculations, specifically talking about the scalability of these calculations to large resources and some of the strategies we have for the portability of these methods, and then maybe some of the future ideas that we could implement with new standards like One API and things like this. Before I get into any specifics, I would just give some high-level overview of what a binding free energy is and why we're interested in calculating it. So in drug design, we might be interested in finding a small molecule which binds non-covalently to a protein, and this protein would be involved in the mechanism of action of some disease, and then by binding to it, we can inhibit its action. So an oversimplification is that this binding affinity is then a proxy for the effectiveness of the drug. So these binding affinities is what TIES calculates, but how do we perform this calculation? So TIES is an alchemical method, and in the computer we can perform alchemy. So we change one set of atoms into another, say maybe A to B, and over the course of this transformation we run molecular dynamic simulations. And the analysis of these simulations can tell us which set of atoms were preferred. So in terms of a favorable binding affinity. So the important thing to understand for the scalability of these calculations is that as we transform from atoms A to B, we do this in discrete steps, and then we call these steps lambda windows. Within each lambda window, we're running repeats of these molecular dynamic simulations, and it's important to run these repeats because the molecular dynamic simulations are going to return a distribution of results. So if we want accurate and precise results, we need to be running replicas. And then the general idea is that these lambda windows and repeat simulations are all independent. And so here already we have two domains over which we can achieve some parallelism. Typically for these calculations, we might have, say, 13 lambda windows and 5 repeat calculations. So just for one binding affinity calculation, this is already 65 maybe nodes or GPUs that we'd be using. So this idea of running the repeats in each alchemical window is kind of what makes TIES unique in the literature. It's not a method I've come up with, it's something that Peter's group have been working on for a long time. Many iterations of this software now. The most recent iteration was done by a previous group member, Matt, and he created TIES 20, and this forms the automatic setup for the protein and the ligands which are used as input to these kind of simulations. So this is what it would look like diagrammatically, where we have TIES 20, which builds the input, and then this is farmed out to the alchemical windows and the replica simulations, which are all in parallel. And then you have some sequential steps, which are for MD type things, equilibration and production simulations and analysis. But since these are sequential, we can't achieve any parallelism there. In terms of portability, our method currently is that since our calculations rely on these molecular dynamics engines, we can exploit the fact that some of them are better designed for CPU or GPU. And essentially, because TIES is a wrapper around these molecular dynamics programs, we can switch between the two different programs at will. And typically, we might use NAMD on a big multi-node CPU machine, but OpenMM on GPU machines with accelerators. But the kind of central idea here is that there's some embarrassingly parallel nature to these calculations, but there are other areas in which we can achieve parallelism to use larger resources. So this is one example of this, where we might be doing some kind of parametric uncertainty quantification, and the results I'm showing here are for a reproducibility study, where we're looking at reproducibility across the two molecular dynamics engines we commonly use, NAMD and OpenMM, but also reproducibility across some analysis methods which are commonly used. So you can imagine here that we have these 65 simulations for each binding affinity calculation, but then we have repeats for the different engines, the different analysis methods, and you can go deeper on this as well. And other members of Peter's group have done more in-depth uncertainty quantification on NAMD, for instance, looking at 127 free parameters which are input to the engine and quantifying uncertainty on this. So the results I show here are just for one protein system with 10 or so ligands, but the whole of the study was 5 protein systems with 55 ligands. So we also get some parallelism there across the ligands, and the general results are, as long as we're running these replica simulations and quantifying the uncertainty, we can get good agreement between the molecular dynamics engines, and we always see very good agreement between the analysis methods. This then is kind of parametric uncertainty quantification, but we can also do an aleatoric uncertainty quantification on these types of calculations. And I already spoke about how, as we run these molecular dynamics simulations, we get a distribution of answers. So here I'm showing the results where we've run large replicas of simulations, so up to 320 here, where typically we might use 5. That's figure A, and then figures B, C, and D are subsets of 32 replicas for these simulations. And the general idea here is that even at the level of 32 replicas, we're still seeing some variance in these distributions. And 32 replicas is 10 times what might typically be used in the literature. So it kind of highlights the importance of these replica simulations. Since we've got so many replicas now, up to 320, it's very easy to use a lot of resources, and the example I've given here is Archer 2, which is where these simulations were run. We ran the simulations in batches of 135, and then this immediately allows us to use almost 1,800 nodes on Archer just in one submission. The final area in which we can achieve some parallelism is what I've called here kind of methodological. So in a drug design campaign, we're looking for maybe one drug or a family of drugs which bind well to the protein, but we have to sort through many other drugs which do not bind well or not at all, or have some other undesirable properties to them. And this, we could be searching through hundreds, thousands, hundreds of thousands. We heard from Austin Clyde yesterday, his screening work, and this is almost 12 million compounds, I think he's looking through that. So there's a lot to search through. A good example of this is fluorine scanning. So this is a methodology where we might take a drug which is close to being at the end of the pipeline of drug discovery or already developed, and then you would systematically swap out every hydrogen or combinations of hydrogens for fluorine. And so you can very quickly generate large numbers of candidate drugs which you might want to test to see if they improve the binding affinity at all. And the example I give here is something we're running on Summit currently, where if we were just looking at, say, 24 fluorinations or fluorinated analog compounds, we're then already using almost 1,600 GPUs on a machine like Summit. To set up these kind of calculations, we've developed the TIAS software, and we're currently in the process of making a new release for this software. So not only does this set up the input, but it also sets up and orchestrates these alchemical windows and replica simulations. And part of the newest release, we have an API now for these TIAS programs. This is an example of what I'm showing here. So in lines 1 to 11, we're performing the work of TIAS 20, setting up the inputs. And then 12 to 13, we run the simulation. 14 to 15, this is doing the analysis. And then you can imagine an external loop around all of this, which would be looping over all of the compounds that you're interested in, or all of the parameters you want to run uncertainty quantification on. So I spoke a little bit about the strategy that we have for portability, which is switching between these codes. And this gives us portability across CPU machines, GPU, AMD, and NVIDIA. But we've heard today and yesterday about these new Intel GPUs and how these are going to feature heavily in upcoming machines like Aurora. So we want to make sure that our methodologies are robust for these new platforms. So part of this new hardware release that Intel is making, they're also bringing out tools, and they've termed these tools and methods kind of in one API. The goal of this is partly to unify programming between GPU and CPU. So on the GPU side of things, they're using a non-proprietary existing standard, which is called SQL 2020. So the question is then, if we want to be as ready as possible to run on these Intel machines when we get access to them, can we take a molecular dynamics code like OpenMM and run this with SQL? So this is what I'm showing here, where OpenMM has CUDA code in it. So can we take a small portion of this CUDA code and run it through Intel's compatibility tools, which they say can convert 95% of CUDA into SQL. So for this small case here, where we've taken just the bonded kernel, which is the part of the code which is responsible for calculating the bonded forces of the system, and we run this through the compatibility tool, then okay, this works fine. Then we get the results and we see similar performance, but there's no optimization on this code, and the cards I'm considering here are not exactly equivalent, but it proves the point that the compatibility tool works. So then can we just take all of the other kernels in OpenMM and run these through the compatibility tool to convert the whole code? Well, no. So what I showed in the previous slide is a simplification. So we've skipped a crucial part of OpenMM, which is runtime compilation. So to achieve their existing level of portability between AMD and NVIDIA cards, OpenMM uses a strategy of runtime compilation, essentially building the kernel at runtime for the specific platform they want to run on. And this is not supported by the compatibility tool. I don't know why this is left out. I know of other codes which also use this strategy for portability. But yeah, it's a shame that it's missing, and it means that it would take significantly more effort for us to make this kind of port. Everything would have to be done by hand. The compatibility tool is not so useful here. So does this mean that we can't run on these Intel machines? Well, as far as we understand currently, it should still be possible to use OpenMM via OpenCL on these upcoming Intel cards, but in terms of how feasible this is or the performance, we still need to get hold of a testbed for this. And we're in the process of applying to machines like Articus, which say that they have these Intel GPUs on. So this is ongoing work still. So yeah, with that, I would conclude and give my thanks to our sponsors and collaborators, specifically the people in British group working on these binding for energy calculations, Shanzu and Agastia and Matt. Thank you for listening.
Thanks, Alex. Okay, so we have time for a quick question. Are there any questions from the line?
Don't seem so. Gerald has still got his hand up, but I think that was previously.
Yeah.
So you were saying that you're questioning whether you can run them on Intel machines. What about the other empty engines, or are they all going down to similar situation?
I think OpenMM is the best shot because it's already got portability and it's using this OpenCL. So as far as I understand, this should work, but some of the other codes have gone the route of just CUDA, and then this may be more tricky.
So you've not tested that?
Yeah.
Just curiosity, how it's a single GPU type of run, and these OpenMM runs, or you have also multi-node, like kind of large?
So OpenMM typically is more geared towards single GPU stuff, but it is possible to do. You just
Yeah,
take the
no,
performance
but
hit.
was more thinking for this kind of testing, like also for addressing like portability, if we would get access also earlier to some test cluster within the GPUs. Like single GPU would be already a good test case for you for that, yes.
Just because of the AMD interest as well, we didn't say much about OpenMM. It just appears to work quite well there, straight off. So there's no particular issue.
No.
We're waiting, as you heard the discussion, doing some time on front end to do things, and maybe Lumi report on in Helsinki. But you were telling me yesterday about the interest of Intel in working with you and us, and he's just told you there's an issue, which if you could facilitate us, we could direct to them and say we do something to help. And
yes, that was
it's
actually indeed the size of the use case to get an idea, because I mean we potentially have access to a test cluster equipped with a Ponte Vecchio GPU. So that could be an easy way to go to install OpenMM and test it there. We have also support from Intel as well to optimize and deploy the code. So that could be a good startup point and scale out also to get bigger systems.
I have a question here. Is it possible like to go through a kind of indirect AMD involving method so that you typify the code? Then I'm not sure if this chip code uses SYCL. Then you would just somehow need to switch between the Intel SYCL runtime or the AMD one.
No, I'm not sure. Not sure. Maybe you can give me the steps afterwards, I think.
Yeah, so I think we can wrap up this first session. The break is a little bit shorter.
No. So what I've said is, so I've contacted Natalia online. She's happy to push it back a little bit. Then we'll have 20 minutes break. We'll come back at 5 to 11. Just gives us a little bit of time to catch up this later this morning. So yeah, 20 minutes now. Yeah. And thank you to all the speakers and Marco for sharing. We'll see everyone online in 20 minutes. So. the two.
Yeah, no, I think this one.
So, did I
Oh, there was one. I think it was. Yeah, there was another blue one. I think. Yeah. Okay.
The CompBioMed conference.
Yeah, that's right.
They're all in bags, so there's.
Yeah, exactly.
I've got about 4 of them.
There's stacks. I'm sitting there thinking, why do I have these things?
You ever need something to write on?
I'll just tell everyone that.
Yes. Hi, Natalia. Can you hear me?
Hello, hi. Yes, I can hear you.
Okay, yes, we are gathering everyone from the coffee break and we start.
Yeah,
So-
I imagine it's difficult to separate them from coffee and attend to the to this presentation.
So so presentation is 15 minutes.
Okay,
But for questions, if it's okay for you,
of course.
I can give you a warning on 5 minutes if you prefer, but
Yeah, fantastic.
15, perfect.
Sounds good.
Okay. I think we can start the second session in the morning. So our next presenter is Natalia Jimenez from Atos. I will present, give an overview and an introduction to the Atos Life Science Center of Excellence. So please, Natalia.
Thank you very much. Can you see my presentation?
Yes.
Okay. Can you see me or not?
Yeah.
Okay, perfect. Perfect. So delighted, well, to join to this CompBioMed all hands call. Unfortunately, I wasn't able to be on site today, unfortunately, because of previous commitments. Well, I'm going to present here the Atos Health and Life Sciences Center of Excellence. I'm not going to make any introduction about Atos because you all know about Atos already after so many years together working in the CompBioMed project. But I think I'm going to give you a very different view of Atos, which is Atos working specifically in this area of knowledge. So basically, a few years ago, let me see if I can move my presentation. Okay, yeah, now, yeah. So, like 3 or so years ago, we decided that we needed to do something about health and life sciences because we realized that we were like manufacturing like leading edge technologies, that we were the #1 in Europe in high performance computing and in worldwide in quantum simulation, but we were still struggling in finding real powerful applications of those technologies, or those technologies solving real problems in the health and life sciences space. So that's we decided to create this Center of Excellence. The center is in South Cambridge. I guess most of you know about the Genome Campus, the Wellcome Genome Campus. Our presence on campus was approved by the board of directors because they look very careful who they admit on campus. We are specifically located at the Biodata Innovation Center. This is a startup incubator. Basically, they are taking companies that are coming from EBI, European Bioinformatics Institute, and also the Sanger Institute. So we are somehow we are like a new guy here in this kind of ecosystem. The office or center is going to look like this. We would like to have like a dynamic kind of flow in our room so that we are welcoming people to come, drop by. You know, you don't need an invite. You are on campus, you drop by, come and see our demos. We are targeting at having 2 demos up and running every time, and we are going obviously to create new demos every time. We have this like brainstorming area where we are going to use this design thinking kind of methodology with the idea of transforming concept into real solutions, POCs, pilots. So that's the idea. So the Center of Excellence at the end of the day is a place to explore, to discover, and to co-create, co-design with people in research centers, universities, pharma, biotech companies, medtech companies, you name them, hospitals, healthcare organizations. So I told you about the office and how it's going to look like. But this is the current status of the office. Unfortunately, I tell you, well, you can imagine that I said that we started discussing about creating the center 3 years ago. But when we were about opening the center, or at least signing the leasing contract with campus, well, the pandemic came, ruined all our plans, and we started operating in a virtual way. So we launched the center virtually back in March 2020. Since then, we've been organizing webinars and courses and all these things. And now I'm super excited because, hopefully next week works in the office are going to finish, so it's going to look lovely. And we are preparing a launching event on the 30th of June, but I'm going to tell you more about that later. Well, just a bit on why Cambridge. Why did we decide to set up this center in Cambridge? This center is something quite unique for Atos, because Atos is a huge IT partner. We are working in, these large HPC deals. Although we are organized in vertical industries, being health and life sciences one of them, but it's the first time that we are really investing in this kind of initiatives. So if you look at this slide on the left-hand side, and these are AstraZeneca headquarters. So they decided to move their research, their R&D, from Sweden to the UK, to Cambridge, to leverage this unique confluence of academia, bio know-how, and medical expertise and experience. And they are located right next to Addenbrooke's Hospital and the Medical Research Council Molecular Biology Laboratories. So AstraZeneca invested over £1 billion to implement this center, which is to increase the level of collaboration and innovative ideas delivered out of that. Well, their share price and pipeline suggest that they are on the right path. Atos is implementing a similar approach to AZ, but much more modest level. And well, these are the two themes that we would like to tackle as part of this center, because we need to find some focus. We cannot address every kind of challenge in health and life sciences, because, as you know, it's a super broad area. But we would like to focus on precision health and drug development. And let me explain a bit about how we are planning to do that. We need to be very humble here, because we know we are an IT company. We don't have a subject matter expertise. So we want to position Atos, or we want Atos to be, for you all, the enabler of the implementation of precision health. If you take precision health as the 4 Ps, predictive, preventative, personalized, and participatory, for the predictive side, we are helping research organizations, hospitals, healthcare organizations to be more efficient in genomic processing and analysis, for example. On the preventative side, we can work in advanced diagnosis, advanced analytics. We can put into play our computer vision kind of expertise with the recent acquisition of companies such as Ipsotech, and also data scientist companies such as Datacentrics. And on the personalized and participatory, is all about the right drug to the right, or the right intervention to the right patient at the right time. Here we have expertise in, solutions that will monitor patients to keep patients at home rather than having them going to the hospital. Also about the drug development that we are going to see later. But precision health is a broader term than precision medicine that is looking at the individual. But here we are looking also at the population level, with predictive modeling, patient stratification strategies, and also the surveillance of new pathogens, now that we are all super aware of a COVID pandemic. Dipping more in detail about this precision health, and the 4 Ps. Our vision is very aligned with CompBioMed, the virtual human, which is basically, we have the virtual human, the fresh and born human being, and is producing big data from the different procedures, interventions related with medical imaging, different modalities of medical imaging, digital pathology, omics technologies, where genomics is only one of them, sensor wearable devices, and also electronic medical records, clinical trials. All this is big data, not because of the volumes of data, but more because of the variety of the data. And what we can offer here is the possibility to speed up those processing analysis kind of methodologies using high performance computing, quantum simulation, AI, IoT, edge computing, you name it. The idea is once you have a digital human, you can deploy any kind of use case on this virtual representation of yourself. And this was about precision health, but what about drug development? In the drug development, well, the thing is that we are in Atos working very closely with pharma companies in the digital twin, which is more looking at the manufacturing processes that can benefit from simulation. And we've been very successful doing that. But we are missing somehow the R&D part of a process and the clinical trials. So from the target identification, the discovery of lead compounds and lead optimization, preclinical and clinical trials. So these 3 kind of building blocks, we would like to explore them further together with users and exploring different use cases where technology can facilitate. The idea is to speed up the drug development, which is currently a painful and lengthy process, so that we can get drug candidates much faster. And well, some kind of use cases, and this is not even a use case, but a reference, because we are, as I said, we are already working with many organizations in the area of genomics, where HPC can be a great accelerator for the processing, but also analytics for once you have the variants and you need to correlate them with what's going on with the patients as part of a patient cohorts. But there are other use cases that are more kind of aspirational use cases for us. We would like, as I mentioned, we have capabilities in computer vision. We would like to deploy them for the sake of speeding up the interpretation of this tedious procedure, which is the endoscopic peel, which will take 1 hour of a highly specialized doctor to go through an 8 hours video and being able to spot what's going on with their patients. And if you see this as an isolated procedure, well, it's okay. But if you have like 8 patients per week and you need to allocate 1 day of your week only being in front of a telly, well, this is very prone to boredom, and so is prone to human mistakes. So if we are able to speed up the process so that in 5 minutes you have like a range of frames that could be like the ones that are anomalous, and you focus all your attention in knowing what's going on in there, well, you are saving valuable time of your medical staff. Another example, and I know that we've been industries have been looking at this for quite a long time already, on skin cancer and remote monitoring of pathologies in the skin of a patients. But I would like to say that this is more on positioning Atos as the end-to-end partner of this kind of processes, because we can do everything in regards to this use case. And to the point of using AI to classify pictures all these things, so that you can stratify patients in different cohorts attending to the risk of suffering skin cancer, and you can play with the predictive side of things as well. And this can apply also to other kind of cancers. And we are also patenting or filing patents, and this is one of them on severe asthma. And you know that patients with severe asthma, they are prone to go to the A&E because of a worsening of their condition. The idea here is to keep patients at home and to keep them monitored. And to do that, the idea around this patent is to blend data coming from electronic medical records of patients, but also weather forecast data, pollen level, pollution levels coming from the meteo agencies, so that you can give patients recommendations through a mobile application, and that is obviously supported by the healthcare system, so that you can recommend increasing the level of inhalers or the dose of inhaler if necessary, or even advice in terms of traveling, you know, or don't walk through this area because it's high in the level of pollen you are allergic to. All these kind of things we can recommend if we are able to create this data lake with all this information coming from different data sources. As I said at the beginning, we are super excited because next Thursday we are opening the center in a physical way. And so we have an agenda, a super excited agenda. We have Pete as our keynote speaker. You are all invited to come to this event. Is from 11:30. We are going to have two sessions, one on precision health, the other one on drug development. There will be plenty of opportunities for networking, and it's going to be great to see people face to face and to interact and to come back to normal. Thank you very much. And if you have any questions, I'm very happy to answer them.
Thank you, Natalia. I don't know if there are any questions from live.
No, I don't see anyone.
Well, one thing more than a question is like the point is like, I think that the interesting things is given also the sometimes difficulty of engaging with hospitals and centers of this size for HPC type of work is, I think it's excellent to see a big player like Atos developing these centers and also I saw like listed hospitals already. So you are really engaging not only with the research part, but really with the MD doctors and hospital sector, and this probably quite difficult to pull in into this digital medicine, but
yeah. Yeah, definitely. So this is our main aim, to make them comfortable with technology, because most of the time it's a problem, it's a cultural problem, so that they don't feel like they are able to use technology. And so I think with the center, we are removing those boundaries, and we are making all these people comfortable with leading edge technologies, because we are providing the physical space for them to have their hands on these technologies. That's the main aim.
Yes. And like, whereas I think it's easier with the AI type of workloads and stuff like that to now start approaching also physics-based modeling, like we do mostly also in CompBioMed, are like going forward quite fast, and yeah, can really see application directly with hospitals.
Yeah.
Okay, I think.
Thank you very much.
Yeah, thank you, Natalia, again. Nice presentation. So we can move to next presenter, another remote presentation from Ioannis Zarrabi. Hope I pronounced it well. It's going to present about porting of MLB to GPU and high performance computers. So Ioannis, you have 15 minutes. please.
15, not 20. Okay.
Yeah.
Okay, so if it's 15, then okay. First, hi everyone. Emily, shall I share my screen or?
Yes, please. Yeah.
Yes. Sorry, that's not the one.
Yep, that looks good.
No, that's not the one. Just a minute to resolve the issue. Okay, I believe it should be okay now, right?
Yeah, that's better.
Okay, so let's then skip to the next slide. So I'm going to discuss about porting HemLP for human scaled up to simulations on GPUs. The outline is the following. First, introduction, covering the motivation behind this work. What is HemLP? Why porting to GPUs? Then briefly talk about the main concepts of the lattice Boltzmann method, which is the method used by HemLP to solve the hydrodynamic equations of motion, which are going to be useful later on for the algorithmic implementation of HemLP and porting the code to GPU architectures. Namely, we developed first the CUDA version, and recently we have ported a code to HEAP with the help of Atos. And recently we're also working on porting the CUDA code to OneAPI, targeting Intel hardware as well. So the introduction, the motivation behind this work is, I guess, obvious. In the area of computational biomedicine, a significant effort is invested into the development of the virtual human, which is a digital replica of an individual's physiology. Creating a virtual human will assist clinicians' to understand how a course of treatment will impact a given individual. And this can be done by simulating the patient using a personalized digital replica. So by running patient-specific simulations and predictive models, we can assist clinicians in how do they proceed with the course of treatment of a given individual. Conducting now simulations for the virtual human will demand codes that can execute and scale efficiently on large-scale computing infrastructures. And this is where this work comes into play. We take one step forward in this direction of creating the virtual human by developing a GPU version of HemLP. And what is HemLP? HemLP is a high-performance parallel lattice Boltzmann-based fluid flow solver for simulating blood flow on patient-specific images obtained from medical scans. It has been developed historically in the group of Peter Coveney at UCL. It is G++ code parallelized using standard MPI communications, optimized for sparse geometries. And with sparse geometries, we refer to situations where the majority of fluid sites are on the majority of lattice points in a bounding box are not fluid sites. And HemLP has demonstrated, the CPU version of HemLP has demonstrated excellent strong scaling performance, up to hundreds of thousands of CPU cores so far on several HPC machines. So we would like to develop a GPU version of the code and maintain these features. These are some of the applications of HemLP. It can be used for investigating blood flow for cerebral aneurysms, magnetic drug targeting, stent design, how surface patterns can affect stresses at the arteries in the region of the aneurysm, retinal vascular flow, coupling HemLP with organs, for example, the heart, and also self-coupling of HemLP with another instance of itself. So, for example, for simulating simultaneously arterial and venous valvular trees. Now, why porting HimLP to GPUs? We would like to take advantage of the presence of accelerators such as GPUs on high-performance computing machines. And if you look here on the 2500 list, in the first 5 machines, 4 of them are equipped with GPUs, NVIDIA GPUs, and AMD GPUs. Frontier and Lumi with AMD GPUs, and Summit and Sierra with NVIDIA GPUs. So we'd like to develop a GPU version of HimLP that is essentially platform agnostic and will enable us to run HimLP on any of these platforms. Initially, we developed a CUDA version of HimLP, and therefore targeting NVIDIA GPUs. Recently, we, with the help of Atos, ported the code to HIP, and therefore we can also target AMD GPUs and NVIDIA GPUs. And recently, we are trying to port the code to Intel's One API that will enable us to also target Intel's hardware, NVIDIA's, and AMD GPUs. So briefly now, some words, some basic concepts on the lattice-Boltzmann method. The equations of motion that we're solving are the continuum and the Narges-Stokes equation, and we're doing this by using the lattice-Boltzmann method. A basic quantity in the lattice-Boltzmann method is the particle distribution functions, discrete velocity particle distribution functions. This can be considered as the density of fluid with velocity I at a given position and time. These particle distribution functions move along the links of the lattice. So here we have a 3-dimensional model with 19 velocity vectors. And these populations move along these directions and collide at the nodes according to rules which impose mass and momentum conservation. Therefore, we recover the continuity and the Narges-Stokes equations at the continuum limit. Now, the evolution of these populations follow 2 steps. First, we have the collision step, followed by a streaming step. Here, we can see that better. So locally, we have a collision step, which basically updates the distribution functions along each of these directions. And then these updated values will stream according to their direction to the next lattice point. For this is shown here on the lower right. We have the collision, and these new populations will then stream at these new sites, as well as incoming populations from neighboring sites will gather at the point in the center. Now, when we have fluid sites next to any type of boundaries, inlets, outlets, or solid surfaces, we need to evaluate that these, after the streaming step, we need to evaluate, for example, if these are the type of boundary, we need to evaluate these incoming populations which are missing, and therefore apply the appropriate boundary conditions. With regards to HimLP, therefore, we have to first initialize these populations to their equilibrium values, based on what is the initial density, initial pressure in the fluid domain, and the initial fluid velocity, and then perform the steps shown here in this rectangle. We have a collision step, followed by a streaming step, applying boundary conditions when appropriate, and then update the time step. At the given frequency, when we require to obtain macroscopic quantities, we can get this from moments of these distribution functions, and this is repeated according to the time steps required. Now, moving on to developing a GPU version of HimLP, we started by developing a CUDA version, and therefore targeting NVIDIA GPUs. And we do so by exporting the compute-intensive parts of HimLP onto the GPU. These refer to the collision step, the streaming step, and the boundary conditions. These are bundled together into a single kernel. With regards now to the types of collision and streaming kernels, with HimLP we have 6 types of collision and streaming. This depends on the type of the fluid sites and their corresponding links to their neighboring sites. So we have fluid sites at the inner domain. These are the yellow ones. We have fluid sites next to walls, the green ones. We have fluid sites next to inlets and outlets, the blue ones, and finally fluid sites with inlets or outlets next to walls. So we developed different GPU collision kernel for these different types of collision and streaming, and the computations are exported on the GPU, complete the collision streaming, and then when required, bring back the data to the host, to the CPU. Now, we must note that one highly efficient way of achieving excellent strong scaling at high core counts is by first doing collision and streaming at domain edges. These are the sites with the darker regions. So as soon as we complete collision and streaming at these domain edges, we issue the MPI exchange, send data to neighboring MPI ranks, and then while this MPI operation is performed, we can proceed with doing the collision and streaming at the inner domain. And by doing so, we can overlap the computations in the inner domain with the MPI exchange. And this has helped us to achieve excellent strong scaling for the CPU version of HimmelsB. Proceeding with the GPU code, we had to make an adjustment in terms of the data layout. So how are the populations, the distribution functions arranged in the GPU global memory, and we take advantage of how GPUs read from global memory. So we go from an array of structure scheme where the data are arranged based on their fluid index to a situation where the particle distribution functions are arranged based on their discrete velocity direction, the lattice Boltzmann index. And by doing so, we have coalesced memory reading from the GPU global memory, and this gives a significant boost in performance. Further optimization to consider in the future is by considering a collected structure of arrays, which is something similar to this, which has been shown to give better results. Another thing that we have applied is to use different CUDA streams, and CUDA streams refer to execution paths on the GPU for all the different GPU operations, the kernels, and the memory copies. And by using these different streams, we can overlap computations, but also can overlap the memory copies from the host to the GPU and vice versa. And by overlapping, we can achieve better performance. The figure on the right is from profiling the code with this with the arteries geometries on Joule's cluster by Dr. Brian Wiley. So you can see the overlapping of the kernels. The figure on the left is by profiling the code using NSAID systems on a personal laptop. You can see after the execution of the CUDA kernel of the domain edges, we have the memory copy device to host, then data are exchanged between MPI ranks, and then once the local rank receives the data from the neighboring ranks, send the data back to the device. So we can overlap completely these memory copies with the main kernel and hide this MPI communication overhead. These are the main simulations domains used for the strong scaling investigation of the code, axis of the legs, the full human Venus tree, which are the blue vessels here, and circle of Willis geometry, increasing domain size as we go from left to right. These are some of the results from testing the CUDA code on Summit, now number 4 in the top 500 list. So with the circle of Willis, we see that we achieved 90% perfect scaling up to 6,000 GPUs, and then continue strong scaling up to 18,000 GPUs. Now the strong scaling behavior drops to 72% around 12,000 GPUs, and 60% at 18,000 GPUs. This can be improved by using a bigger simulation domain, which is labeled as ExaPipe, which has 37 billion sites. So it has improved, as you can see, it has improved the scaling behavior just because we are increasing the computation to communication ratio. comparing now the CPU and the GPU versions of HimLB, we utilize the same simulation domain, set of Willis. Open boxes are simulation results on Summit, on Supremo Genji. Field symbols are the results on Summit on NVIDIA V100 GPUs. So we see that we observe almost 2 orders of magnitude speed up. So the GPU version of this one was roughly around 300 megaloops per CPU or GPU core. The CPU version of the code is running at roughly 300 megaloops. We must note that these results are with an older version of our CUDA code. A newer version has demonstrated an additional speed up of a factor of almost 2. These optimizations I'm referring to were by focusing on the main GPU collision kernel by performing loop unrolling using pragma_unroll 19 for the specific stencil of the lattice box method we are using, by doing also loop merging, and also interestingly by increasing the number of registers per thread. So by compiling with this flag, we allow for more registers to be used per thread, and these obviously we had some spilling to GPU global memory. This reduced this unnecessary memory traffic and increased the performance of the code. So, for example, the stalled warps have decreased by 82%. The arithmetic intensity increased from 0.32 to 0.73. This is the compute weight over the data movement. For compute-bound algorithms, we are in this region of the roofline analysis chart. And we tested that on Summit, for example, with the V100 GPUs. The performance went from 285 up to 550 megaloops per MPI task. Now, porting the CUDA version of HimLB to heap, this was done by Atos, who ported the CUDA code to heap. So we can now run HimLB on AMD's GPUs. So the code has been heapified. So what is heap? Heap is AMD's dedicated GPU programming environment for producing high-performance kernels to execute on GPU hardware. It can also be used for portability solution with already written applications in CUDA. So by providing a source-to-source translator to convert from CUDA to heap. There are 2 approaches to heapify CUDA code. One is the heapify Perl, and the other one is the heapify CLang. Atos followed the second path given the complex structure of HimLB. So this also has a dependency with CUDA. And the heapified version it is easy to follow the syntax of the heapified code as basically the terminology changes from CUDA to heap. So we have, for example, for errors handling, we have heap error instead of CUDA error. For the different streams and synchronization points, CUDA stream synchronize or heap stream synchronize. Launching the kernel, so it's easy to go from using now the heap API. It's easy to understand the changes in the code and then perform further changes. And we must know that the CUDA kernels haven't been changed at all. So they can be used as they are. Now, with when compiling the code, we can specify which target, what is the target hardware. So whether it's an AMD or an NVIDIA GPU, by using these terms here, Initial results from Atos on 3 different clusters have been performed. Okay, it's not a straightforward task to compare because we have different CPUs, different network, different software stack and Linux distribution. Maybe based on the cost per socket, we can say that the V100 GPU is closer to the MI50. And these are the results on a small simulation domain. The V100 GPU and the MI50 show similar simulation times. These are on a bigger simulation domain with the MI100 and the V100 GPUs. What is important here is to test the heapified code on targeting the same hardware. So we tested that on Summit. So we tested the HIP-UEFI code versus the QDAC code, and we observed similar performance, which is something very important to ensure that there is no bugs or any overhead with the HIP API. And finally, here I'm gonna discussing about the One API, porting the QDAC code to One API. What is One API? This is a set of tools and libraries for developing high-performance applications across diverse architectures. So it's a C++. It features a C++ compiler that implements syncall. Now the porting process is it's something really straightforward. Intel developed this compatibility tool, DPCT, so you can convert the QDAC code to DPC++ source code, SQL code, and by using this tool, it transfers almost 90% of the code, and then produces this human readable code within like comments. For example, here produces these comments about it's complaining about this QDA status, which is of type QDA error. And nevertheless, it did transform the memory copy to SQL. And then once we manually fix any errors, we can compile. We can also choose the target hardware. Here, for example, we're targeting an NVIDIA GPU. So what we did is to first try by porting a single QDA collision kernel, manually fix the errors, and then test on, we did the testing on CSD3 at Cambridge. The native QDA code shows better performance compared to the ported code. I guess this is to be expected at these initial stages. And the next thing we did is to try to port the full HIMLB QDA code. This is a bit more tricky. We need first to generate the JSON file, which will include which has the include paths and macros definitions, and then run the compatibility tool with this file, which then produces the converted code. And we're now in the fun part of manually fixing any errors and try to compile the code and test. So to summarize, HIMLB is a numerical code based on the lattice-Boltzmann method. We developed the GPU version of HIMLB based on QDA. This has demonstrated excellent strong scaling performance up to tens of thousands of NVIDIA GPUs. We are now in the process of porting to One API. We did port the code to HIP. So we'd like to have a platform agnostic code to target a wide range of machines accelerated by GPUs. So at this point, I would like to thank you for your attention, and I'm open to any questions.
Thanks, Ioannis. See if there are any questions from the online attendees. Doesn't seem to be.
I have a question about the model itself. So is gravity something which has to be considered in such material flow simulation?
Sorry, was that question about gravity? So within the framework of lattice-Boltzmann method, you can impose body forces. This, but yeah, whatever I have shown is doesn't have any of these. But I mean, for example, the magnetic targeting paper I showed in the beginning, this was with implementing body forces in the code as well.
The obvious, from Ioannis's point of view, going back to what was said also by Alex earlier, is we're interested in accessing very large machines with GPUs that may be AMD, common Intel versions. And so Intel's not yet here,
Yes.
but we have connections in this project I'm looking at you because
Yes.
that's the same thing. And
Yeah.
then AMD, well, Frontier suddenly arrived.
Yes. For the AMD, we may have a more official structure rules to have access to Euro HPC on me. We have to check
Yeah.
also well there. And the Frontier. Intel for the large scale, I'm not sure how large would be the access.
That's the question.
Yeah.
But in Leonardo, for hearing, I was talking over coffee with a bit more detail on, Sapphire Rapids things.
Yeah.
Leonardo, he's optimistic that it might be in production soonish, early next year,
Yeah.
dependent on a number of things.
But this is the CPU part.
No, I agree, but
Yeah.
that's still interesting to us. And then there's the conversion to the PVC.
Yeah. GPUs are the NVIDIA, right? The A100. Yeah. So that we can already have some sort of scalability.
I mean, this is classic case that if you are willing to do testing on the system. So if you are willing to test on the system and try to scale up, we can set it up.
We are very willing to do it. Yes.
But do you know if you will, because now Euro HPC has these benchmark access codes for really actually benchmarking software on, especially on Lumi, and if Leonardo will be included one of the system when in production, this benchmark.
We are following the same
Okay.
The same principles. So 50% of the production will follow the same path for access of
The Euro HPC.
of the units.
No, good, because that could be something already, maybe preparatory discussion with you guys if possible, and then apply for what is access. Okay, Yannis, thank you very much for the presentation. I think should move to the next speaker.
Thanks, Marco.
Thanks, Yannis, again. So, Sharp Law from UCL, also talking about imposing boundary condition with blood flow simulation and MLB. So, Sharp, please.
Thank you for the introduction. So I'm going to present you a recent study on imposing boundary conditions on the outlets of a large arterial network. In addition to myself, this work is contributed by John and Peter. So the creation of the virtual human is one of the goals in CompBioMed. This requires the development to capture the human biology at all scales. And today I'm going to bring us closer to achieving simulation of blood flow at the full human scale. Since in the audience, you may be already familiar with HemLB, so I will skip the introduction of this. So what I'm going to talk about is how we impose the boundary conditions. Due to some constraints in the resources, sometimes the simulation domain is truncated and you just focus on the domain of interest. But this requires some accurate representation of truncated systems, as shown in the gray area in the left figure. And this the strategy to impose boundary conditions must be efficient and scalable, being able to scale up to a large arterial network. So common strategies include imposing the pressure or the traction at the boundaries. More sophisticated strategies include imposing the flow rate plus some assumptions on the flow profile. And a rather accurate method is to couple the main solver with reduced order models. But this requires some parameters tuning in order to have an accurate representation of the truncated system. So the right figure shows an example of such a coupling system in which at the three outlets, there's a circuit or reduced order model attached to it, and there are some parameters that you should tune it. So Greenberg and Kaniadakis proposed an efficient strategy to impose the flow rate ratios at the outlets. The relation in this strategy is that the flow rate is inversely proportional to the resistance in the reduced order model. So because it requires only one parameter, this can be scaled up to a large arterial network. So they show that this model is accurate if the resistances in this model are sufficiently high, as shown in the results in the right figure. So here our contributions is to clarify the general role of the model parameters in this strategy, namely the resistance and the capacitance in this reduced order model. We study the impacts of these parameters on the stability, accuracy, convergence of the simulations by varying the model parameters using like a parametric analysis. So to give you a better understanding of this strategy, I'll first go to the rather basic concept of this. So in fact, the blood vessels can be conceived as an electric circuit. The pressure can be is analogous to the voltage, flow rate versus electric current, flow resistance versus resistance, wall elasticity versus the capacitance, flow inertia versus inductance. So imagine the whole human circulatory system can be conceived as just a large electric circuit. That's amazing. So this Greenberg strategy relies on the rather simple or relies on the two-element Wing-Kessel model, which is simply a RC circuit. So the dynamics of this RC circuit is given in the formula on the left. And this model basically converts the flow velocity, which is given by the main solver, to the pressure, which is required by this solver. This model has the advantage of being able to filter high-frequency noise, which often hinders the simulation unstable. So I'm going to present how we approach this problem instead of just going to the results, because I think it's very crucial. And but however, in the next few minutes, it may be a bit intensive. So in this Greenberg strategy, there's a knowledge gap in which we don't know how large the resistance should be. They just say that, we just know that the resistance should be high enough, but how large should it be? So first, for an arbitrary vessel, we can conceive it as a cylinder of a length L and radius R, such that the flow resistance is the same. Then the flow resistance is given by the product L times K, where K is given by the Poisson flow. So in the case of outlets, the total resistance, the vessel is attached to the two-element Wing-Kessel model. So the total resistance of this system is given by the sum Lk plus R. So in Greenberg's relation, there's no dependence on this product Lk. So this suggests that a necessary condition for Greenberg's strategy to hold must be that the resistance dominates the product Lk. So as I mentioned, we don't know how large the resistance should be. So we use a parametric approach, and we introduce the parameter gamma R, which is like the scaling factor of this Lk. And we will vary this parameter in a range. Okay, similarly, for the other model parameter, capacitance, we do some analysis. Back to the dynamics of the two-element Wing-Kessel model, the RC model, we find the homogeneous or transient solution. And the characteristic decay time is given by the product RC. In the bottom plot is the time series of the pressure. We see that there's some transient solution, which is some noise, and it is very it's unphysical, and we want to avoid this. So in order to avoid this, the characteristic time for it to decay is given by RC. So this RC shouldn't be too large. So we have this upper limit for the capacitance. where omega 0 here is the fundamental frequency. You can find it by like the period of the inlet profile. So for the lower limit of this capacitance, we find it from the Fourier transform of the partial differential equation. And we find that the pressure and the flow rate is actually has a phase difference given by the arctangent of omega RC. And this can be translated to a time lag in the time series of pressure and velocity, as shown in the left figure. You see the peak of these two profiles has indeed some time lag in the experiments. So this gives the lower limit for the capacitance. And all in all, the upper limit and the lower limit suggest a parameterization for the capacitance, which is this formula. You can see another scaling factor, gamma RC, is introduced here, and the resistance is put on the left right-hand side because it's given by the previous parameterization. Okay, this is the last slide about the strategy we use, which is about the desired flow rate ratios. So based on the principle of minimum work, we show that the flow rate is proportional to the equivalent radius of a vessel. With the mass conservation, this relation can be written as this form. So the significance of this relation is that we only need the information about the geometry, but not some data on the flow. This is particularly useful when the physiological data is unavailable. However, some studies show that this relation is not accurate in some conditions. For a more accurate description for a particular case, other powers other than 3 can be used. Okay, so I've already finished the most intensive part, and I'll now show some results in this study. First of all is the simulation domain. We use 2 domains, namely the 5 outlets domain and the profunda femoris. And the latter of which is a model for it's an artery in the right thigh of a human. Is the name of that artery. So we look at the stability of the simulations. Here I plot the parameters RC against R, and the green spots represent the simulations in which is stable, and the cross means unstable. So we find that the stability increases with the value of the capacitance, which is given by the boundary of the 2 regions, the blue line here. And we also observe that the stability increases slightly when the grid resolution increases. This is expected because it can resolve more features in the flow. Regarding the accuracy of this model, we plot the simulated flow rate ratios against the desired flow rate ratios. So as the resistance increases, namely the color becomes deeper, we find that the plot approach the dotted line. Just the dotted line, which is the expected one, and the error decreases exponentially as this resistance increases. At the largest resistance tested, the error is within 7%. However, we also we observe some intrinsic errors, namely that the error doesn't approach 0 as the resistance increases. This may be explained by the fact that the simulation is not completely convergent. And this is about the impact of the capacitance on the accuracy. So we find that there's only a slight dependency of the capacitance in this aspect. This is also expected from the relation of Greenberg's relation, because in the relation, there is no dependency on the capacitance. Okay, the next one is about the convergence and the fluctuations of the simulation. Here shows the time series of the flow rate against the flow rate at different outlets. So as the capacitance increases from the left to the right, we can see there are more fluctuations and the convergence rate decreases. So this is expected because the capacitance is related to the storage property, which can reduce the instability and also is related to the phase of the flow. Okay, so in addition to what I said, the time lag is also observed when the capacitance is increased. So Greenberg's strategy is an attractive approach in which we can use it to for a large arterial system. And here we use a parametric analysis to understand what practically the values of the parameters should be. We find that the value of the resistance should be larger than the threshold value. And we propose an estimation of this threshold value, which I didn't go into detail, but it's tested and shown in here. Another aspect is about the capacitance. I show that the capacitance doesn't have a strong dependency on the accuracy of this strategy, but it has impacts on the convergence rate and the fluctuation of the flow variables. Also, the simulation stability is also impacted by this parameter. As the capacitance increases, the stability increases. So our understanding here can provide a guideline for choosing these two parameters in practice. And the methods used here can be applicable to larger and more complex vascular domains encountered at full human scale. This study wouldn't have been completed without the support from these parties. So this is the end of my presentation. Thank you for listening.
Thank you. So, are there any questions from the online audience?
Can I ask a question? This is Alberto.
Yes, please, Alberto.
Hi, Sharp. Hi, everybody. Very interesting and useful study, Sharp. Thank you. I have a question on the way that, or not, it's a curiosity really, whether you impose any constraint on the total impedance. You've identified some ranges for the individual parameters of the Wing-Kessel models that might lead to instabilities. But in the community, often what we do when we impose these Wing-Kessel models, we look at the change of impedance, and we impose a constant impedance when we go from a higher dimensionality model to a Wing-Kessel model. And this is to avoid reflections at that, let's say, interface. Do you do anything like that? So do you also impose that sort of constraint on the overall impedance of your Wing-Kessel parameters?
The overall impedance, I didn't impose it, and yes, I just vary the value of the resistance.
Yes, so basically you're developing a strategy on how to choose those parameters. One of the things that you might want to look at is this change in impedance, because if you have a change of impedance across that interface, then you might risk generating artificial reflections that shouldn't be there. So this is another thing. Yeah. Okay. Thank you.
Thank you.
I understand correctly, you solve an OD density, which you then kind of prescribe as a boundary condition in the lattice plot.
So we use the boundary condition already developed in MLB. So it assumes that there's a, so it interpolates the density at the boundaries to the lattice point next to it. And so because the pressure is just proportional to the density, so we don't have to solve an ODE.
But like the solvers, so for this Windcastle model and the CFD simulation, are they run with the same time step?
Yes, so it updates every time step.
And the second question is like related to this instability. So is this instability like originating this Windcastle model or from the CFD simulation?
I think it's the other way around. Without this Windcastle model, the simulation becomes more unstable. This Windcastle model encounters for the phase differences between different outlets. So, so there will be less high frequency waves, which impede the stability of the simulation.
Yeah, but is it not maybe like that these waves could be, so if you had, let's say, higher precision, if you did all the computations in precision, would that prevent the instability, or it's really a physical instability mechanism?
So if you have patient-specific data where you know the exact phase difference at the outlets, then it should be fine. But in this study, we don't use patient-specific data. We rely on the calculation from this Windcastle model. Yes. So if you don't have the right phase difference, then in each iteration, or it will generate a high frequency wave. Then of course you can make the simulation more, denser. I mean, the resolution can be increased very high, but it's not efficient. But it has a very high computational cost.
Thank you, Shah.
Thank you.
Again. And we can move to the next presentation. So it's a remote one. Emma, can you hear us?
Yes, I can hear you. Let me just share my screen.
Great. So next presenter is Emma Fine from Atos, presenting the work done in a co-design between Atos, ARM, and UCL on a genomic application. Whenever you're ready, you have 15 minutes plus some time for questions.
Okay, thank you very much. So today I will be presenting about life science workflows, ARM-based platforms. And as you mentioned, this is done with Atos in the Life Science Center of Excellence, as Natalia mentioned before. And then it was also done in collaboration with ARM, University College London, and the University of Oxford. So what do I mean by life science workflows? Well, commonly in the HPC world, we see GROMACS, which helps with high performance molecular dynamics. But the one I'll be discussing today is a new tool in the life sciences called Viridian. It's a virus assembly tool. So what it does is it takes in different fragments of DNA and it assembles them to try and get the whole picture of the genome. And then afterwards, it checks different frequencies to be able to help detect mutations in SARS-CoV-2. And this was developed by the Sam Iqbal lab at the University of Oxford. So the DNA fragments that I was discussing before, amplicon reads, are from Illumina and Nanopore. And Viridian itself has a couple different dependencies like Minimap2 and Raycon, PySam. So what we were able to do is we were able to install a lot of these dependencies, and we used SPAC to do that. And then also with the work of Dr. Alex Wade, who also presented earlier, he was able to also really optimize. There was a very large for loop in Viridian, and he was able to get the quality control section down from 70 seconds to 10 seconds. So we also worked with the development team to optimize Viridian itself. And excuse me if I lose my voice, I'm recovering from COVID. So yeah, so the objectives of this are really, there are two key important things. In the life sciences and high performance computing, traditionally, life science workflows have mostly or always been run on x86 platforms. And the second thing is that there's a growing need for cloud-based platforms in the life sciences to help with collaboration. And so that leads us to our question, which was, are life science workflows efficient on ARM-based platforms? So with our partnership with ARM, we were able to access the ARM Neoverse-based platform, the Ampere Ultra, which is a cloud-based platform on-site at Atos. And we were wondering, how does this platform compare to other x86 platforms that are available at Atos? And we wanted to know the comparison of the performance of this at the core, socket, and node level. So here's a brief comparison. We see some major differences in the number of sockets. For example, the Ampere Ultra is 1 socket. We see differences in the core levels. Ampere Ultra is 80 cores. Intel is 32 cores per socket with 2 sockets. AMD is 64 cores per socket with 2 sockets. We also see differences in the CPU speed, so the frequencies. An important thing to mention is, well, x86 frequency have turbo and boost mode that can be enabled. The Ampere Ultra always runs at 3 gigahertz. And then we also see differences in the processor subsystems and the memory. These are all 8 memory channels. I'll leave a little bit of time so you can get in all those differences. Okay, so now on to the results. Oh wait, sorry. I guess I'll talk a little bit about what we did. So what we did is we launched N multiple instances of Viridian. This was sequential and single-threaded. So what we did is we would launch a Viridian. One run of Viridian takes about 2 minutes to process, and so we would run it on 1 core. When it was done running, it would run the next instance of Viridian, and so our whole goal was to fill up the socket and node level and core level, and then we measured wall time in order to accurately compare these. So here are some of the results we got. So for the first comparison of the performance on the core level, we ran 1 run, 1 core, and this is the wall time we got. So what's interesting is we do see the Ampere Ultra is on par with Intel and AMD. It's not extremely slower, which is something that's good to know for it as a tool. And what we did next is we normalized these wall times based on the frequency. And so here are those results. So next we wanted to compare on socket level. So what we did is we decided to run 320 runs of Viridian. Why did we choose 320? Well, we just chose the least common multiple of all the different number of cores on each platform. And here we see that the Ampere Ultra performs extremely well on the socket level. And we normalize these wall times as well based on the frequency. So yeah. Next is we looked a little bit at cluster time. So with turbo and boost enabled, AMD and Intel still perform better than Ampere Ultra in terms of baseline As you can see, it's right in the middle. And next, we also had a look at wall time in comparison to the number of cores. And we do see that AMD and Intel are still slightly faster, but the Ampere Ultra, again, is in the same range, which is nice to know because it's not traditionally used for life science workflows. And finally, we decided to look at 640 runs on one node. So the same idea was to fill up the entire node. So we just doubled the number of runs that we did on the socket because x86 platforms have 2 sockets. And again, you're then able to normalize that, and these are those results. So the main takeaways are that ARM-based platforms performed on par with x86 platforms. The other thing is that we were able to run Viridian and work with that development team. So that's a big step in the life sciences area with helping detect mutations in SARS-CoV-2, all the different variants. And then we also saw that ARM-based platforms performed well on the socket level, which could make them a new emerging tool in the life sciences. And the next step is that Atos was able to access the 128-core Ampere Ultra as well. Those results have not been done yet, but that's the next step in the process. And thank you very much to CompBioMed, of course. A big thank you to Alex Wade at UCL for his work on the optimization within the loop of Viridian. Of course, Peter Kovney from ARM side, Phil Ridley and Daniele Piccarozzi, as well as the ARM HPC user group. A big thank you as well to Sam's lab, who developed Viridian, and at Atos, huge thank you to Xavier, Opa, Erwan, and David for all the technical advice on a lot of this project, and to Natalia, who we saw present before in the Center of Life Sciences. Thank you.
Thank you, Emma. Thank you. Any questions? Maybe first online. No, not the case. Any questions here?
I think Natalia is on. Can I ask her indirectly? For your launch on the 30th, is this something you're interested in telling people? I can't remember. You have Intel, you have ARM people attending. Can you hear me?
I'm sorry, it's a little bit hard to
speak to Natalia actually, just to check if she's there.
Yes.
Do you want? Is this something that matters to you in a launch context? Because you might be saying something about the virtues of Intel versus ARM, or is it not something to discuss?
Yeah, definitely. So far, it's not part of the agenda, but we can feature that as a demo. That would be very nice. Yeah, I don't know, as you are there, if you are able to deliver on that, because I'm afraid that for Emma, because of her being recovering from COVID, I don't know if it's going to be possible for her to come. But definitely, yes.
I mentioned it, but my question to you, I hope your understanding is, it's you've got 2 competitors, vendors. You want to set them up against each other there? I mean, Intel's been so standard in bioinformatics. This is sort of suggesting that ARM can be competitive in that space. Is that, what does Atos think about that? Because it doesn't care.
We are agnostic, so we do not care about that. I mean, we are going to do whatever is best for our users and customers. So we are not going to get married with any kind of technology at all. So this competition, I think, is very healthy, and is going to help us all to reach to the right solution for the user.
Okay.
Yeah, I think just to add on what Natalia said, of course, all the platforms I think in Autos's level are useful, and so it's just whatever people prefer with their different programs. And the thing is, then on the life sciences, it has been usually done through x86 platforms. So it's just interesting to see that other platforms could be used.
Yeah, and for the opening, we've invited all of them. I mean, Intel, AMD, ARM people. We don't know yet who is going to come, because some of them have a register, but we don't know for sure if they are coming. But definitely everyone is invited, and not only in terms of processor, but also storage, NetApp, DDN, and other partners, cloud vendors. Everyone is invited. I mean, we want to be part, for them all to be part of an ecosystem, and we all collaborating, because I think there is a place for everyone here.
Emma, before I have one question about your presentation. Mostly, did you do any tuning on the installation of? Because I saw one of your first slides. You were mentioned that you were using SPAC probably to install the different components of this Viridian workflow. For the ARM-based runs, did you do any specific optimization for the installation? There is anything like peculiar to ARM to configure, or is just a basic installation?
Yeah, so I would say that the installation of Viridian, we did have some difficulties, but I think it was more on the Viridian side in terms of there weren't certain dependencies listed, but otherwise for the installation on the platform, it seemed to go fine after using SPAC. Yeah.
It does provide SCMD data validations. It enables this. Okay. So on Intel, indeed, we did vectorize, enable vectorization, of course, but ARM also.
Okay. Good to know, because I think these are the good information also at the beginning, like to understand where to start, especially for new people. And but thank you.
For this processor, it doesn't do SPE vectorization, just like Mark used yesterday. It does different kind of
like this
SCMD instead of SPE.
Yesterday was mentioning also the length of the array, 256.
Yeah, this is 128.
Okay. Even okay. Super. Thanks. I don't think there are questions. So thanks again, Emma, for the presentation, and thanks for the speaker. Morning again. I think we are
ahead of time.
Ahead of time.
Well done.
So, well.
Yeah, the clock on here is wrong.
Everybody's happy that we have 1 minute more. Yeah, 5 minutes more for time.
Yeah,
lunch.
Lovely, thanks, Mark.
Yeah, thank you. And I think we come back at 1:25 as anticipated for the work package presentation.
Yeah, and just to say that the next part of the all hands meeting is for core partners only. Yeah, Terry said, can I show the timetable? I'll put that up before I go for lunch, but my computer's the other side. But yeah, thank you everyone, and enjoy your lunch, and we shall see you back soon.
Emily, can you hear me?
Yes, I can hear you.
Oh, hello.
You're going to the
Hello.
Oh.
So that works. Oh, I need to do that system preferences thing.
Why not do this instead of Alistair? I don't know how it works. Well, you should. Yeah. I don't know that it's nice.
Hello.
Hello.
Should work now. um, Why isn't that showing up? Ah, there it is. See that all right?
Are you sharing?
Can't say today.
Oh, I am sharing. Oh, hang on. Hang on.
Okay.
I'm asking for more permissions.
Thank you.
Hello.
Hello.
Oh, let's just tick that now. It says it's sharing.
Let me hang on. I'll just get my laptop. Sharing on.
Yeah, nothing.
Let me try it once more because I just activated a thingy.
Yes, I would definitely be ready.
It'd be quite good to I could watch football and I could do that.
Good to be said.
Emily, if there's struggles, I could just send you the slides. You could do it.
Yeah. There we go. Looks like
Oh,
yes.
there we go.
Oh, there you go. Right, let me see if I can do full screen. All right.
Yeah, there we go.
Woohoo.
That looks all right.
Good.
Good. Excellent.
May as well leave it up there if that's okay.
Yeah, sure. Yeah, no problem.
We'll be starting in a few minutes anyway.
Yeah. Cool.
Yes. Okay, so I make that 1:25. I think we've got some people on the line, and hopefully some other people from here will be joining back again. They're just outside finishing lunch. So I think we get started on the work package 1 presentation. This will be done partly by me here in Bologna, and then partly by Hugh back in the UK. And Hugh has control of my slides, so could you move to the next one? Thank you. So yeah, I think there's a lot of people who are aware, but for those who are not, I will be leaving UCL and therefore CompBioMed as project manager from beginning of August 2022. Just want to say that I've really enjoyed working with everybody, and it's been a great experience. Nick Laver, who is in the room, but you guys can't see it back home, will be taking over with some support from CBK and the work that they do, or we do. So yes, that will be from August, but just a quick update on some of the work that we've been doing. Again, project meetings as usual taking place, Focus COE and CSA liaison. So Focus COE came to an end, I think early this year, and will be we don't know how that will continue exactly, but the HPC3 meetings are still ongoing, and still get involved in those, and that's quite a useful part of actually getting things done as part of the COEs. In terms of periodic reporting, there will be another reporting period at the end of September 2022, so this year, and we're looking at organising the review meeting for December 2022 to give us time to to give you time to write the report and to get that submitted before the review meeting, which just makes it a little bit easier for everyone. KPIs, we still need to keep an eye on those. That's something that will need to be reported at the end of September, so we need to make sure that we're heading in the right direction with those. And the deliverables have all been sort of submitted in time and everything. So could we move to the next slide? So yeah, deliverables, as I've said, for this year we've got 6 coming up. So that's month 36, 37, and 38. 2 for each of those months, sort of spread out a little bit so that they're not all clumped together. I think from what I understand, they're all in good order, and people are prepared to start work on those. And just to make you aware that at the last review that we had with the Commission, they sort of applauded us for putting the deliverables online, but asked us to only do so after they'd been approved by the Commission. So the ones from last time that we needed to update still haven't been approved, so they are not available on the website, but if you need them, we have them available. So yeah, next slide, please. And then milestones, again, linked into the deliverables above, and quite confident that we're in a good way for delivering those or for reaching those. So I think that is the end of my part of the talk, Hugh. Yes.
I think so. All right. Does that look all good?
Mm-hmm.
And you can hear me. It does sound like it. All right, so I'll jump in and cover the dissemination and innovation aspects of Work Package 1. So a year ago at the last AHM, I was discussing how we had to adapt our strategy to meet the needs of the height of the pandemic, really. And once again, we